Good day, ladies and gentlemen. I'm Dr. Mohamed Baha Saeed, expert on microbial biopesticide. Today, I will present a scientific paper about the biological control of Tuta absoluta in Saudi Arabia, promising strategy using entomopathogens. This work is part of research development project in organic agriculture funded by Ministry of Environment, Water and Agriculture in Saudi Arabia and implemented by the Saudi Organic Farming Association. This project was hosted at the National Organic Agriculture Center. We will start this presentation with focusing on the justification for conducting this research. It started with Tuta absoluta can cause up to 100% crop losses if left untreated, and also there is need for applicable microbial biopesticide focus or targeting on the larval stage of Tuta absoluta in the soil. Also, the soil itself can provide protection for the targeted entomopathogens and protect them from the harmful environmental condition, and by this way increase or enhance their efficacy. Now we need to have a look about the objectives of this study. The main objective of this study was the controlling Tuta absoluta by targeting on the soil stage with the indigenous microbial strains. Also, the second objective, we need to check or evaluate the heterohabitus indica efficacy and be very Bayesian efficacy against the soil dwelling stage of Tuta absoluta, exactly focusing on the larvae. Also, the last objectives, we need to study this effectiveness of sequential application of these targeted microbes. Before we go in details, we need to have a look about the life cycle of the Tuta absoluta, which is the targeted best in this study. The life cycle starts when female lays eggs on the host plant. The larval stage causes the greatest damage, and under optimal condition, larvae pupate in the soil. After about 9 days, an adult emerges and ready to mate and invest new plants. Therefore, our study focused on targeting and disrupting the soil stages to effectively break the life cycle of this pest. Also, it is good to have an idea about the life cycle of entomopathogenic nematodes, which is one of the targeted microbes in this study. Entomopathogenic nematode infect insect host through natural opening and release symbiotic bacteria that rapidly degrade internal tissues. This process kills the insects within a few days and allow the nematodes to feed and develop and produce inside the cadavers or reproduce inside the cadavers by the thousands juveniles, then emerge to seek new hosts. Due to this efficient mode of action, these nematodes are highly effective to managing soil-based stages, which is the Tuta absoluta larvae in this case. Also, similarly, entomopathogenic fungi kills insects through a sequence of events that includes spore adhesion, germination, penetration, and spreading inside the host. And this process typically takes 3 to 5 days, depending on environmental condition and developmental stage of the targeted pest. Now in this section, we need to present a simplified sequence of events that illustrate the key stages of conducting these experiments, which was started with preparing the seedling of tomato plants and then planting these seedlings. After that, rearing the Tuta absoluta adult stage and release them on the tomato plants to start their infestation. After that, back to the laboratory to prepare the entomopathogens and apply them on against the targeted pest in greenhouse. After that, monitoring, collecting data, and analysis the results. Uh, Also, it is important to provide an overview of the experimental design used in this study, which was structured as the randomized complete block design with four replicates. Here it is clear we use the single applications and sequential applications to have a look on the sequential application uh, process against the targeted pests. Now, in the results, we need to focus at the beginning on the single application. It is clear that when you use any of the targeted microbes singly against the larvae of the soil of the Tuta absoluta in the soil, the mortality was less than the 50% for both strains, Beauveria bassiana and Heterohaptitis indica. But the scenario was changed when we moved to the sequential application. Here, it's clear that the larval mortality increased and the best treatment was applying Beauveria bassiana followed by nematode after 24 hours. These treatments produce the highest effectiveness, which was almost 65% against the larvae in the soil. Also, these charts provide a clear overview of all treatments together, showing the sequential application, as we can see, outperformed the single application as we discussed in the last slides. Also, this image demonstrates the ability of local or indigenous strains of Heterohaptitis indica and Beauveria bassiana and showed their rules against the targeted pest. Here it is very clear these larvae killed by the Heterohaptitis indica and these larvae killed by Beauveria bassiana, and here this is clear, healthy, untreated larvae. These highlights their potential and as a safe and effective options for disrupting the Tuta absoluta larvae in the soil. Okay, now we can conclude from this study the following points. The sequential applications outperform the single application as we see in the last slides. Also, applying strain NOAC N7, which was the Beauveria bassiana, followed by strain NOAC N8, which was the Heterohaptitis indica after 24 hours, resulted in almost 55 percent mortality against the soil dwelling larvae of Tuta absoluta. And when we applied them individually, nematode showed higher efficacy compared to Beauveria bassiana. At the end, the recommendation of this study, we need to optimize the sequential application strategy to first maximize the larval mortality and second to suppress the adults stage emergence. And by this way, we can reduce the load on against the healthy plants. Also, implement field trials under diverse conditions in Saudi Arabia to validate the consistency and effectiveness of these treatments. Here, I focused exactly on the sequential application. Also, advance the development of integrated microbial formulation, or we need to find formulation that allow us to use the Beauveria bassiana and Heterohabitus indica together to be easier for the farmers for application and to be after easier to give us very good results. By this recommendation, now we can say thank you for listening and see you in another event. Thank you so much.